อยู่ชายทะเลบางขุนเทียนอาชีพทำวังกุ้งครับทำมาหากินก็อยู่กับพ่อแม่ตั้งแต่เล็กมาอยู่ในปัจจุบันนี่ก็ประมาณ60 ปีนะตั้งแต่เกิดนะไม่มีป่าชายเลนนะมันก็คนพื้นที่มันก็อยู่ยากอะเมื่อ10 ปีที่แล้วป่าชายเลนมันสมบูรณ์จะหาปูหาอะไรเนี่ยมันก็มีเยอะเดี๋ยวนี้ว่าคลื่นเซาะมาเงี้ยตลิ่งพังหรือว่าพื้นที่ชาวเกษตรเนี่ยล่นมาเยอะคนละหลาย10 ไร่มันพังเข้ามาก็ต้องล่นหนีกัน
Mangroves are very important for coastal regions and estuaries where they provide erosion protection against surf and storm damage, and they provide nurseries for young fish and crabs to replenish fish stocks in the ocean.
There are two main ecosystems for Thailand. Fishery depends on mangrove forests, coral reefs as well. So if we don't protect these two ecosystems, eventually it's going to affect our fishery and affects the quality of life in general. Over the past 50 years, I think more than 60% of the mangrove forests were destroyed, and the conservation programs and the restoration programs are carried out without considering genetic factors or genetic diversity.
So it's important to see how much genetic diversity they have, because if the genetic diversity gets too low, then the populations are at risk of collapse.
So 5 years ago, we were approached by the Department of Marine and Coastal Resources. They are trying to protect the mangrove forest and maintain the genetic diversity among the species that we study. So we tried to obtain the reference genome sequence for 15 mangrove species, and we're expanding that database as well. Since we obtained the genome database, we are able to assess the level of genetic diversity more accurately using the DNA data instead of morphological evaluation. There are some really rare species that can only be found in Thailand. And we did some of the first genome sequencing for the prevalent species in our country, like the Bulgaria as well as the Cereops.
Sequencing is very necessary for our research. Using techniques like RAS-seq, we can identify and study specific genetic markers in mangroves. We apply the RAS-seq technology using MGI to our mangrove projects, as it provides sufficient SNP data per sample for our diverse biosystem study.
We also use MGI-seq to do RNA sequencing, and that is to try to understand the gene expression required for some of the mangrove species to be able to grow in salt water, what sort of genes or pathways have to be turned on for them to be able to grow in such a high level of sodium.
I think so our MGI technology have the strength point about the higher precision, good technology, and as well as reasonable price to support this project. We are a reliable partner to contribute the Nation Omic Center to do the mangrove forest project.
I think the technology is going really fast. The cost of sequencing has come down a lot. So we can do like a larger study. I hope we can continue to use MGI technologies to do a lot more study with mangrove forests, with some of the other projects that we have in lab.
And we wanted to do restriction enzyme sequencing so that we can identify how much genetic variation exists across the populations all over Thailand. So from the Chao Phraya River outlet all the way down to the south.
People, they weren't really aware which species are going to become extinct first because they didn't really know the level of genetic diversity at the DNA level. So our studies actually review that information, and so it sort of prioritize which species to save first.
I think by building up a database of reference genomes, that we do have a bright future for mangrove conservation.
We're also doing an outreach program where we educate the general population on why it's important to look at genetic diversity in mangroves. And hopefully by doing that, they will become more aware of the importance of mangrove forests, and they would feel like protecting the forest for future generations.